Yeah! Once again, it's the return of the prodigal daddy here to talk at you about muscles, or should I say muscles. Once again, it's the end of a weekend. That means no shave, no hair product, and we're ready to talk. We're talking about the microscopic anatomy of muscles, and then we're going to get into more details about how they work. Let me show you like this. I can't make a music video as well as I did before, but that's okay. Skeletal muscle cells are multinucleated. These nuclei are just below the sarcolemma here that you can see, and they look like a zit. I feel like I just want to pop that, but it might get all over you, so I won't do that. Here you see 1, 2, 3 nuclei in one muscle cell. The sarcolemma is kind of the equivalent to the periosteum, where we saw was the connective tissue wrapping. Here we have connective tissue wrapping around the muscle. So the sarcolemma, as I said, is a specialized plasma membrane of the muscle cells. And inside that, there's one organelle that's different from what we normally see, and that's called the sarcoplasmic reticulum. And this is a type of smooth ER, or smooth endoplasmic reticulum, and you'll see later that it stores calcium. All right, so here we have the sarcoplasmic reticulum. It's the purplish blue thing that's being shown over here. We'll talk about the yellow stuff here in just a second. These are T tubules. You'll notice we're focusing in down on one myofibril of a muscle fiber or muscle cell. So this is even smaller than one cell. These are the parts within a cell. And you'll notice each myofibril here gets wrapped by the sarcolemma, by the sarcoplasmic reticulum, and also the T tubules that you can see there. Now, to give you some idea of what we're talking about here, when the muscle contracts, pieces inside the muscle have to move towards one another or shorten. And in order to do that, there's all kinds of things in the way to prevent it from happening. So normally your muscle is at rest. Now, to begin, these T tubules are invaginations, I know what you're thinking, that is a real word, of the membrane, the sarcolemma, going down into the muscle, and carrying with it the ability to transmit a message. So when a neuron synapses with this sarcolemma, and then the sarcolemma plunges down into the muscle, it carries the impulse or the message from the nerve or neuron with it down into the muscle. And as it passes down through these T tubules, they're called, they pass right next to this sarcoplasmic reticulum, and that triggers the sarcoplasmic reticulum to release calcium. And the calcium is going to enable the muscle to contract. Now, within the myofiber, or muscle cell as it's called, there are myofibrils. Within so to give you some idea, each one of these things is a myofibril. So 1 myofibril, 2 myofibrils, 3, 4, 5 being pictured here. Within a myofibril, we have bundles of myofilaments. Okay, and the arrangement of these myofilaments, specifically actin and myosin, give each myofibril a distinct pattern or coloration, segments of light and dark. And the light band we call the I band, and the dark band we call the A band. And you can start to see those here. Here's the A band. This region here where you see red is the dark band, and this region where you see the blue is the I band, or the light one. So what we have is one contractile unit within a myofibril, and again, remember, a myofibril is the smaller part of a myofiber, and the contractile unit is from Z disc to Z disc. Here you can see this jagged line right there. I was going to do this as if that would help you understand it, but maybe it does. From Z disc to Z disc, that's one sarcomere, and that's the region that's going to shorten. Now later we're going to get questions on, what does the Z disc do? Well, the Z disc is a place for actin to attach. This blue stuff is actin, and the red stuff is myosin. So thick filaments, as they're called, are myosin, here shown in red. And the thin filaments we'll talk about are in blue. Those are called actin. And the myosin has ATPase enzymes. Now, any word that ends in ASE is an enzyme. And ATP is an energy molecule. So the important thing here is when I have myosin here, when ATP binds to the myosin, that ATP immediately breaks apart into two things, ADP, or adenosine diphosphate, and a phosphate group. So the bond that was holding one phosphate, two phosphates, three phosphates, one of those bonds breaks and you lose that phosphate. But the important thing there is that bond was a high energy bond, and when you break that bond, it releases energy. And that energy is that which is going to let you shorten the muscle. So the enzyme that causes ATP to break down or to break apart and release energy is actually the myosin, the thick filament itself. Then the thin filaments are the actin, as I mentioned. Now, here looking even closer in, here you see myosin in the middle, and these things that look like golf clubs sticking out, or punching bags, or scrotums, if you like, awkwardly pointing up or down. Those are what are going to bind to the actin, or the thin filament. And those heads of myosin, the myosin heads, are going to move like this, and we'll show you exactly how that works. But when they move, they pinch like this, and they shorten or pull the actin filaments in. So here I have actin, here I have a myosin head. It attaches and moves in this way, pulling the actin in or shortening it. What you're going to notice is that the thin and thick filaments overlap, and there is a region where they don't overlap, mainly at rest, and this is called the Bayer zone, or actually also the H zone that we'll talk about later. Now, some generic terms here. Irritability is the ability to receive and then to respond to a stimulus, and contractility is shortening in response to that stimulus. So here we see skeletal muscle is innervated by a motor neuron. To innervate means to be connected to, and a motor neuron is a neuron that is going to try to get you to move, to perform some motor function or motor action. This is the spinal cord, and here the neuron is coming out of the spinal cord and synapsing with muscle cells. Now, one motor unit, as we call it, is just one neuron and the muscle cells that it stimulates. Now you notice one motor unit is going to stimulate more than just one muscle cell. Here we have motor unit 2 stimulating 1, 2, 3 muscle cells all at the same time, whereas motor unit 1 stimulates a different subset of muscle cells. So the point where the neuron meets the muscle we call the neuromuscular junction. Funny how they came up with that term. And this is the place where a message is going to travel across, and that is where we have what we call a synapse. And we'll get into synapses more in detail later when we do the nervous system. That's the next unit. But in general, we do need to have a basic understanding of that. That's kind of just regular bio things. So here you see the end of the neuron, okay, and here's the muscle. Here you see these T tubules that I mentioned, and here you see the myosin and actin part within a sarcomere, within a myofibril, and then you can see other things within cells like mitochondria and so on. So the synaptic cleft or synapse is the gap between the nerve and the muscle. They actually don't physically touch. There's a little bit of a space, and that space is filled with interstitial fluid, and that fluid is going to enable things to pass across. Now, how do they pass across? Well, there's something called the neurotransmitter, and I'm going to go back here for a second. Here you see these little balls here. These are called vesicles, and inside of them is neurotransmitter. And when calcium enters the neuron, it's triggered to enter. Those details are for another chapter, but when it's triggered to enter, this vesicle is going to bind to the membrane and release these little tiny blue dots. I think it's blue, unless my eyes are failing me. Those blue dots are neurotransmitter, and in the case of the neuromuscular junction, it's the neurotransmitter acetylcholine, or ACH, and that's the neurotransmitter passing across. It's stimulatory, so the neuron is going to, using ACH or acetylcholine, it's going to bind to the outside of the muscle, to the sarcolemma, and trigger an impulse to continue through the sarcolemma, through the use of sodium. So sodium is actually what enters into the muscle to cause the message to continue passing through the muscle and through the T tubules. And that sodium rush, that sodium entrance, generates or causes an action potential. An action potential is a complicated term, really complicated, 2 words, which just means impulse or message being sent from one area to the other. And it triggers Ka plus to be released from the SR, or sarcoplasmic reticulum. And just so you know that when your muscle starts to shorten, there is no stopping it. Now, of course, I can shorten my muscle part way. I don't have to do it all the way to complete shortening. But that one sarcomere from Z disc to Z disc When it starts to shorten, it's going to go all the way. There is no partial and then letting go or stopping. It goes all the way. Starts. Once it starts, it goes. Can't be stopped. We call this now, when we do this moving, the shortening from Z disc to Z disc, the sliding filament theory of muscle contraction. It is just a theory. It's the best we've got. It's our current understanding. We can't actually visualize and see what's going on, so this is the best understanding that we have. So I mentioned that Ka plus was released from the sarcoplasmic reticulum, and when that happens, that causes the myosin heads to bind to actin. Now, you can see the little golf club heads a little bit here on the myosin, and these are the myosin heads. Here in blue is the actin. And when these myosin heads are going to bind to the actin, only when calcium is around, we'll show you later why, that's going to cause the muscle to shorten. And you notice the H zone is going to disappear when fully contracted. Okay. Myosin heads then bind to the next site in the thin filament. So essentially, if this is my thin filament and this is my myosin, this is a binding site, this is a binding site, this is a binding site, you're going to have binding, shorten, release, bind, shorten, release, bind, shorten, release. And that's how it works, of course, much faster than that. It's boom, boom, boom. Obviously, you couldn't see that because it was so fast. And then, so that's how actin slides past myosin. And then the muscle is shortened or contracted. So here we see this picture kind of showing in detail what we're talking about. I'm going to talk about what this purple thing is here in just a second. It's called troponin, and this other violet thing is called tropomyosin. But basically, this is actin. Actually, this violet thing is not tropomyosin. It's actin, but I'll get to that in just a second. Here we have the myosin golf clubs, if you like, the cross bridges or the heads. And when calcium is around, here we see Ka2 plus release, these little red dots. And that moves this purple thing, and that's called troponin. And that opens up binding sites. So these are kind of the lock. And calcium is the key. It unlocks it and causes it to move. And now the myosin head can bind to that binding site on actin. They can physically create the attachment. And then the attachment is going to relax back to normal and cause the actin, the thin filament, to move, which is what you can see here. And we'll talk about how the ATP works in just a second with an animation. So this is kind of the overall big picture. Now, the next thing I want to show you is an animation. Now, this is maybe one of the best animations I've ever seen on this topic. It doesn't have sound, so I'm going to walk you through it, and it does take some 3, 4, 5 minutes, so hopefully that's okay. But you can watch through it. So we're narrowing down in on the knee, and it's showing you the generic anatomy of the knee. Oh, my knee is getting sore, actually. And so here you see the muscle. Here's the patella, as you know, the tendon, and we're going to focus down in on a muscle. Here's the muscle, and we're going to look at one muscle fiber within. Here's a fascicle. Here's a muscle fiber. We're going to zoom down in on that muscle fiber or muscle cell. And here within you can see myofibrils wrapped with sarcolemma, and you can see multiple nuclei, of course, right? And you can also see the stripy patterned appearance. Here you see the dark region and the light region, and they'll go through that. The red is myosin, and the, I guess that's brown, is actin. And so a sarcomere is from Z disc to Z disc. Now there's also an M line, and this is the attachment point for the myosin, whereas the Z disc was the attachment point for the the actin. Okay, the A band is only where you see myosin. That's the dark region, and the H zone, as you can see here, is the lighter section in the middle of the A band, and the I band is the light region, shown here, where only actin is found, no myosin. So it looks a little bit lighter because myosin is thick, makes it dark. Now zooming in on those structures, here we see actin, and if I pause it, I don't need to pause it. This thing that looks like purple poop is troponin, and it covers up the holes, the binding sites for the myosin. And this blue thing I call like a bike chain, helps to cover up these binding sites. This is tropomyosin. Now here comes calcium. When that calcium enters from the sarcoplasmic reticulum, it's going to bind to troponin, and that's going to cause troponin to move and unwrap or unhide, if you like, these